So we ended taking a look at how our Fisher and our Haworth projections looked. Remember, Fisher projections are normally used for our open chain form, but we know that if we have a solution of, say, glucose, that what normally happens is that we have the cyclic form forming and then reopening and then reforming. And so we end up with basically a 50-50% of beta and alpha cyclic forms. It's called a pyranose form because it looks like pyrin, the molecule pyrin. It has a 6-membered ring. And which carbon is the anomeric carbon in our numbering system here? Number 1, that's right. And the way you looked at it is to determine that was you looked at where the dangly carbon was, carbon number 6, right? The one that's not in part of the cyclic structure. And if that's pointing up, then you look to the oxygen, and then the carbon to the right of the oxygen is going to be the anomeric carbon. What do I mean by anomeric carbon? What was it before? Yes, it was the carbonyl. It was the carbonyl, C double bond O. And before, when it was in the open chain form, it was not chiral. Is it chiral now in the cyclic form? Yeah, because we have 4 different groups. So that's another one. And so we tell the reader where the OH is, because there's a 50-50 chance if the OH is pointing up or pointing down. And birds are in the air, so when the OH is pointing up, we have the, we call it beta. And then the D stands for the carbon number 6 pointing up in our form. Or if it's carbon number 5, and you look to the right, if that's the oxygen, then that 6 is going to be pointing up in the way we write it.
So if the big group on 6 was down where hydrogen is, it would be L?
Not L. We're not doing L or D. We're doing beta and alpha. Okay, for this one, we're not, we're delineating, because it's a cyclic form, we're not going to do D or L. All right, the D or L, all right, is all of these, we're only dealing with D-glucose now. And we know that it's D-glucose because this carbon number 6 is pointing up. If it were L-glucose, the carbon number 6 would be pointing down. Yeah, so you'll never see that, all right? So when we're looking at the anomeric carbon, the notation is alpha or beta, not D or L. Very important, all right? So it's beta if it's pointing up. If it's alpha, if it's pointing down. Now I just want to point out ribose is a 5 carbon, so it is a pentose. Very good. It's a pentose. So again, number 5 is then going to be the dangly carbon. And so we take a look to the right of that. If the dangly carbon's pointing up, we look to the right, the oxygen. And so carbon number 1 is going to be the anomeric carbon. Now we normally write the abbreviated Haworth. So we are going to just leave in the abbreviated form, leave out the hydrogens. So again, we're going to remember our form for our Fisher projection. This carbon number 1 used to be the carbonyl. So if you look at the carbon after that, OH is going to be on the right, left, right, and then right. Remember, that's how we're going to remember our structure of glucose. And that's fair game for the test on Friday, the structure of glucose. Because we're going to spend a lot of time the rest of the semester on glucose. So I want you to remember that.
Sorry, can you go back for a second? So you're saying we're always going to have D, but the A or B is based on the OH on the first carbon?
The biologically active form of glucose is the D-glucose. So from now on, all we're going to see is D-glucose. And the way we know that it's D-glucose is we're going to write, if I said write D-glucose, and I don't say in the Haworth projection, what I would do is do glucose is 6 carbons. 1, 2, 3, 4, 5, 6 carbons. I know you guys can't see it, but the first one is an aldohexose. So you know that about glucose. So an aldo means that it's So the aldohexose, hexose means it's 6. Aldo means this one is the carbonyl. Then I said right, left, right, right, right? And then the other one is just CH2OH. There's no direction to it because it's not chiral. So that's how you're going to remember. And then you're going to fill in with hydrogens to make 4 bonds to each of them. I won't get you if you do an abbreviated one. Okay? But that's how you'll remember how to draw the structure. All right. Now let's talk about reactions. If that anomeric carbon is free, what do I mean by free? See that on the OH, this one is, what form is this? Is this alpha or beta? Alpha, okay. So we have an alpha D-glucose here. And if, it doesn't matter if it's alpha or beta, but if you look at the oxygen, the OH, that is on the carbon number 1, or the anomeric carbon, that's the carbon number 1, the anomeric carbon, and you see an OH on it, and you see the H on it, that means it's a free sugar. It's not in an anomeric bond, a glycosidic bond. So if that is free, then we can say it's a reducing sugar. What happens is that there's a reaction that's going on here. What is happening to this carbon number 1 from here to here? This structure to this structure. Double bonds forming. So what does that mean to this structure, to this carbon? Is it being oxidized or reduced? Oxidized, right? So what's happening, since we have a hydrogen on the oxygen, this carbon can be oxidized. And because this can be oxidized, you don't just have an oxidation. You have to have a reduction with an oxidation. So what it's going to do is it's going to reduce the silver on this complex. And when it reduces it, it goes from silver on a +1 charge to silver 0. Well, that's metal, just solid metal. So what you see is make a silver mirror on your test tube. Did anybody ever do this in a biochem or a biology lab to see if you have a reducing sugar?
We did a different test.
Did the other test?
Yeah, that's what the Benedict's one.
Yeah.
Sounds more interesting.
So this is really important that we can identify which anomeric carbon is free, which is not in a glycosidic bond. We're going to talk about glycosidic bonds in one moment. Not the first carbon, the anomeric carbon, because if it was a ketose, then the double bond would be here. And so if it's a ketose, then the double bond would be on the second carbon. And so if it's on the second carbon, then there would be another carbon on there. I'll point that out in a moment. So if it's involved in what we say a glycosidic linkage, there will be a negative tolerance test reagent. If another anomeric carbon is free, there'll be a positive tolerance test. And the positive is that you'll see a silver mirror because the silver ion is going to go from a silver ion to silver metal. Okay. Now, so they can be oxidized. When we oxidize the top carbon, then we call it an aldonic acid. When we oxidize the bottom carbon to an acid, we get a uronic acid. So top is aldonic, bottom is uronic, and both is alderic. That's just some definitions. So let's go over that again. So we have, so here we have glucose, and what we've done is this is normally the aldehyde in carbon number 1. So what we've done is we've made it an acid. So that's an oxidation, going from an aldehyde to an acid. So where we go, we call it an aldonic acid. When we've taken a monosaccharide and oxidized the top carbon, carbon number 1. When we've oxidized the terminal carbon, carbon number 6, it's called a uronic acid. And when both the top and the bottom, the aldehyde and the carbon, the terminal alcohol has been oxidized, then that's alderic. So there's some definition words that you need to know. We can also reduce them. And one of the most important reductions is where we've taken out an OH on this carbon number 2 on ribose. Remember, ribose is an aldopentose, and there's got to be an OH on the on every single carbon, and except for the one that's an aldehyde. So here we've seen that we've gotten rid of the OH here. So that's why we call it deoxyribose in DNA. We've gotten rid of, we've reduced an alcohol to just a hydrocarbon. And if you're chewing gum right now, then you probably can look on the ingredients and find sorbitol or xylitol as the non-sugar sweetener in your sugar, in your gum. So that's something we do. And so that by reducing that aldehyde, what we've done to an alcohol, it keeps the sweet taste to it, but it's not a sugar anymore. And so we've reduced those carbon number 1s. Okay, what we're going to see, so we can oxidize, we can reduce these sugars, and we can actually add groups to it. So one of the first reaction that happens to a glucose when it enters the cell is that we pop a phosphate onto it. It requires energy. What we're going to do is break up an ATP, which is it's our energy currency. That holds free energy. And so what we're going to do is we're going to have a nucleophilic substitution where we're going to create a single bond between the oxygen of our dangly carbon. Remember I said carbon number 6 is dangling out there? And that means that it's dangling for a reason so that it's sterically available to do some reacting. So this oxygen is available and it's sterically able to attack the phosphate of an ATP. There's a lot of tension in these phosphates that are right next to each other. So what we're going to do is form a bond, and then we've phosphorylated. So this is a phosphorylation, and so we've activated this glucose by adding a phosphate group. This keeps the glucose in the cell. It stops it sufficiently charged enough that it's not going to get back through the permease enzyme. So it's going to stay within the cell at this point. But we're going to study this reaction next chapter when we take a look at glycolysis. It requires energy. And, of course, it's controlled by an enzyme. And this is called a phosphate ester here. All right, so I've alluded to this before, but probably the most important reaction that we're going to talk about this semester, this chapter, is forming a glycosidic bond. A glycoside is where we've taken the OH on this anomeric carbon, the OH that's dangling off of it, and we've replaced the hydrogen with some carbon. Now, the carbon can be in simple alcohol. It can be something crazy, and we're going to see how crazy it gets later on. But it can get really, really crazy. But we can see that this was now a reducing sugar, and since we have a carbon here, it is no longer a reducing sugar. So to tell whether you have a glycosidic bond or not, you take a look at your sugar. You find your anomeric carbon. How do you find your anomeric carbon? You look at the oxygen in your ring structure As long as the CH2OH, the dangly carbon, is pointing up, then you look to the right. You guys need me to say that again? You look for the oxygen. To find your anomeric carbon, you look at the oxygen in your ring. You look to the left of it. If the carbon number 6 or whatever carbon that is hanging off of your ring structure is pointing up, then you're going to look to the right, and that will be your anomeric carbon. Remember how we made this, right? We had the tree fall down and the back end of the tree curl up, and this is carbon number 5 that had the oxygen that nucleophilically attacked the carbonyl. Yes?
Will you ever see that dangling carbon coming down?
If I flip it over like a pancake, when we look at our disaccharides, you're actually going to see a situation where in order to draw it properly, it's going to be pointing down.
And then in that case, would you go?
You have to really think about it.
Okay.
You have to use your imagination and flip it over, or maybe turn your head upside down. So to deal with the case that the CH2OH is not pointing up when you look for the oxygen in the ring, and it's not pointing up, then you just flip it over in your brain. I'm not going to do that to you on an exam, all right? Especially not this first one. All right, so we're able to find the anomeric carbon. Once you find that anomeric carbon, you find whether you have an alpha or beta OH. Now, if your anomeric carbon is not carbon number 1, if you have a ketose instead of an aldose, you've got to make sure that you're looking at the right OH. Because we could have a case if this was a hexose, but it made a 5-membered ring because it was a ketose, then there could be a CH2OH pointing up here, and it still be a. Let me. I don't want to confuse you guys too much, but if I had this case, this sugar, and this was just a CH2OH, and this was my aldehyde. I'm not my aldehyde, my ketone. It would. So this one would attack this carbon. All right? And so you would have a 5-membered ring. This would still be your anomeric carbon.
Dr. Barrett, it's kind of hard to see.
Is it?
Yeah. Well, it's actually impossible to see.
Let me draw it here for the thing to see, and then I will go and draw it above. So we can see that if the carbon that is the anomeric carbon is carbon number 2, then there might be a CH2OH there. But this is still a free anomeric carbon because we find the anomeric carbon. You look for the OH that's attached to it. It doesn't matter if there's another CH2OH. All right, let me draw it over here. So we have a ketose. 1, 2, 3, 4, 5, 6. A ketose means that we have a ketone instead of an aldehyde. So to make the ring structure, this oxygen is going to attack that one. So what you have is a 5-membered ring. That's the oxygen. This is carbon number 6. This is the anomeric carbon right here. To the right. So if this is the anomeric carbon, you're going to have a CH2OH on it, pointing up or down. And this is an OH. So here, this is this carbon. This is that oxygen right here that did the attacking. This is the anomeric carbon. You guys with me? So I want to point out that this is still a reducing sugar because the OH off of the anomeric carbon has an H on it. So it's a free, we call it a free anomeric carbon. Because it's a ketose, the anomeric carbon is going to be the second carbon, not the first carbon. I need you guys to understand that the anomeric carbon doesn't have to be carbon number 1. On an aldose, it's carbon number 1. Because this one is going to attack here and make okay, I'm not going to draw the rest of them, but this is going to be then the carbon number 1. So I need you guys to understand that the anomeric carbon does not have to be carbon number 1. It's going to be whatever carbon was the carbonyl. Okay, so tell me, if it's an aldose, the anomeric carbon will be carbon number 1 or 2? 1. If it's a ketose, it'll be carbon number 2. Ketoses are always going to be the second carbon. There's no like ketone in the middle. Okay. All right. So what I wanted to point out was that there could be a CH2OH here. All right. There could be that. But because this is free, that's telling me that it's free to react. But we don't have that here. All right, that would be a 7-carbon chain. Okay, so what happens in forming a glycosidic bond is we're forming, what is the form here? What is the functional group that we're forming here? We have a carbon, oxygen, hydrogen, so this is an alcohol. And we're condensing it with another alcohol to form a what? Esters have a carbonyl. Ether. That's an ether. Very important for you guys to know, it's an ether. It's almost like a double ether because we have carbon, oxygen, carbon here. So that's an ether. And then we have carbon, oxygen, carbon here. Now, it doesn't just have to be a CH3. It can be any number of things. We're going to see that it's going to be another sugar ring. When you have the condensation, putting together of 2 alcohols, one being the OH on a anomeric carbon, and then the other being whatever you want. Whatever you want. Okay? Just another OH. Anything. If there's no hydrogen on the OH that is on the anomeric carbon, then you have a glycoside. So I'm going to give you a structure, and I'm going to ask you to circle the anomeric bond. The glycosidic bond, I'm sorry. I might ask you to point out the anomeric carbon and to circle the glycosidic bond.
Is the test going to be the same format?
Yeah. The test is going to be the same format, multiple choice and short answer. Okay.
We don't have to know the whole reaction.
You don't have to draw the whole reaction. What I'm going to do in this test is show you a structure. But you should be able to do it at this point. This isn't that bad. You're going to look and find that OH on the anomeric carbon and put together. You take away the hydrogen and you put together the group that's not the OH there. All right. So this is how we're going to link our sugars in disaccharides and polysaccharides. So here, if you look at this sugar, this monosaccharide here, you can see that this is an alpha, right, because it's pointing down. And it gets a little crazy here. An alpha because it's pointing down. And then it's not going to the anomeric carbon on this one. It's connecting to the 4th carbon on this glucose. So here, we have an alpha 1,4 glycosidic bond. So this is how we're going to notate our glycosidic bonds. We're going to first say, is the OH on the sugar that has the anomeric carbon in the glycosidic bond, is that alpha or beta? So when you're naming these polysaccharides, disaccharides, when you're naming the bond, you first identify the sugar that is in the glycosidic bond. You find out whether the OH is pointing down or up, and that's what this alpha has to do with. So this is alpha because this is pointing down. And then you number it. First you number the carbon that is the anomeric carbon on the first sugar, and then you number the carbon that the OH has bonded to. So here, this is carbon number 1 on this sugar, and this is carbon number 1, 2, 3, 4 on this one. So this is an alpha 1,4 glycosidic bond. Sure. So what you're going to do is first, in any to identify what kind of glycosidic bond you have, you first find the sugar that is in the glycosidic bond, that has the anomeric carbon involved in the glycosidic bond. So find that sugar. Second, you look and see whether that is an alpha or beta. So this one's pointing down, sort of, kind of. It's pointing down, so we're going to call that alpha. Then you number from the sugar, you find the carbon, you number the carbons that are involved in that glycosidic bond. You're going to first number the one that has the anomeric carbon involved, so that's carbon number 1 in this case, and then the other sugar. So this is alpha 1,4.
How do we know the other one's beta?
This one, we don't know if this is, this could be alpha or beta. It doesn't matter. We don't know anything about this one. The linkage here, saying alpha 1,4, says nothing about this OH, whether it's up or down. Doesn't matter.
I guess I'm just confused on the second one because I would say.
Oh, I haven't gotten there yet. Let's just focus. Is everybody good on this one? Yeah? Okay.
Explain how you numbered the one to the right.
This one? Okay, so first I found where the anomeric carbon was, and I found whether this is going to be carbon number 1 or 2. This is going to be carbon number 1 because I don't see another CH on that one. So this is carbon number 1, 2, 3, 4. Just remember our tree going down. It's all going to come back to the tree. Don't ever cut down trees by yourself. Okay? We're just going to do it in our brain. Here, we're going to cut down the tree of our sugar. All right, let's move on to this one. All right, because this is from the textbook, and they wanted to conserve space. Okay, they're trying to say, okay, let's look for the one that the anomeric carbon is in. So we say this one because this is the anomeric carbon here. That's not in the glycosidic bond. So this one is the one that's in the glycosidic bond because we know that this is the anomeric carbon. All right, so this is their feeble attempt when they go bloop like that it was alpha because it's pointing down. Really what we should have done in space is push this up here. Okay, to draw this better, it would have been better to have this up here. All right, so here, if this were up here, and so they would be pointing down, so this would be alpha, because this is pointing down. It's our feeble attempt. Does everybody see that with a little J there? So it's pointing down alpha. Oh, that's carbon number 1. What is it connected to on the other carbon? Carbon number 1, 2, 3, 4, 5, 6. So this is an alpha 1,6. Okay, you guys with me?
And as far as naming, that's all we need to know is like alpha 1.
Yeah, you got to name the bond. To name the bond. Yeah, there's other stuff I'm going to ask you to know. All right, now, this one's nuttiness. All right, this is actually wrong. What I want you to do is to correct this. This should just be down. Okay, so if you guys can correct that. There's no bloop there. This is just down. And we're going to say alpha down there. So there's two corrections. I could have corrected either way. All right, so if this is just down and it's connected to this one, then what do you notice that's different from up here? This one, up, our anomeric carbon is involved, right? So that's going to be alpha. Oh, wait a second. In my other one, the anomeric carbon's involved. Oh. So I have to say beta. You could do beta alpha, alpha beta. Doesn't matter. You pick one, right? You pick one. So then that one's beta. This is quite the awkward way to write this, right? Like that.
So you could call it alpha or beta.
No,
Or beta.
this one, yeah, the order doesn't matter right here to talk about the linkage, right? So this one has to be called alpha because it's pointing down, the way I wrote it. This one has to be beta because the way I wrote it up here. And then it's connected at both in carbon number ones. So what they did to make it look better is this is the case where I flipped this one over like a pancake. So from here to here, it's flipped. Because you see the dangly carbon? Can you guys do that? I find that some of the younger people cannot do this because they didn't play with G.I. Joe guys when they were little. They played with video games. Right? Imagination. You have to be able to like imagine things. So here, this one and this one are written the same, correct? But this one, I have it pointing down, but that's because this and this, it's just flipped over. Okay. Can you just recognize that? All right. Do you think I'm going to do that to you on the test?
So how do you know if it's beta? Is it because both the
No, here, I know that this is beta because look at this one. This is at the anomeric carbon. It's involved in the glycosidic bond, and it's pointed up. Right? Up means beta, down means alpha. All together like this. I would probably show you something like this or this to say what kind of bond do we have. All right. So no, I wouldn't show you them independently. This, I'm not going to do this one to you, but I would do this to you. I'm not going to have one where I flipped it over. So this is how we're going to put our sugars together to form disaccharides and polysaccharides. The important monosaccharides that I want you to eventually get is our glucose, our galactose, and our fructose. Oops. Our glucose, galactose, and our fructose. The glucose is the one that you are going to be immediately responsible for in this exam.
Dr. Bear,
Yes.
I have a question. For the slide 23, why are we not also specifying D in the name?
We're just naming the bond. I'm not naming the whole thing.
Okay.
I'm just naming the bond here. So yes, you would, to name it properly, you would say D-glucose or whatever. Yes.
So for the beta, it doesn't necessarily always have to involve the anomeric carbon for the second ring.
For the beta, always has to be on the anomeric carbon. But the anomeric carbon doesn't have to be the first carbon.
Oh, okay.
Okay? Like I showed here, that in here, the anomeric carbon doesn't have to be the first carbon. If it's a ketose, then it's going to be the second carbon. But that means that it's going to be, the first carbon is going to be dangling off. I haven't really showed in my slides that example. I just want to caution you. So here, up on here, we only had one anomeric carbon involved in the glycosidic bond. But in this case, both of them were. Now, I'm going to do a little foreshadowing. This one is, and these, which the top or the bottom would be a reducing sugar. The top or the bottom, which one will be a reducing sugar? What do you have to be a reducing sugar? You have to have a free anomeric carbon. The top. Yep, these are our free, quote-unquote, free anomeric carbons because the anomeric carbon has the OH on it. There's an H on the O that's bound to it. This is tough, guys, if you're taking vacations in your heads. And I can see by your faces a large population of you are off somewhere nice. Spring break is coming. Okay? Spring break is coming. So here, and this is the free anomeric carbon. Yes?
So that makes it a reducing sugar.
That makes it a reducing sugar because it can get oxidized there. Okay. So again, these are the, did I answer your question?
Yeah.
Yeah? Okay. These are the sugars that I eventually want you to know the structures of. We're going to come back and look at those later. Glucose, I do want you to know. Fructose, look at that one. This is our example of a ketohexose. This is the anomeric carbon. I found the oxygen in the ring, made sure that the dangly carbon up to the left of it was pointed up. And then here. How did I know that it wasn't flipped over, that this was not the dangly carbon? There's got an OH on it. Very good, Sean. There's an OH on it. The carbon number 6, all right, is going to be pointed up off of the carbon that doesn't have an extra OH because this was the OH on it, right? Remember this. This is carbon number 5 in our structure. Carbon number 5 in our structure. The OH on the carbon number 5 attached to the carbonyl. So the carbon number 5 can't have another OH on it. This is it. So this is carbon number 1, 2, 3, 4, 5, 6. It might take some marinating for you guys. I knew that this was the anomeric carbon because there's both the CH2 and an OH. This is not the anomeric carbon because there's just an H on it. There's no OH on it. Both of the right and left off of this oxygen have a CH2. Which one is the anomeric one? It's going to be the one with the OH. Do you guys see that? Okay. A little tricky there. Fun with sugar. All right. And amino sugars. Oftentimes we're going to do some, and with sugars, it gets really crazy what happens with sugars. We're just going to do crazy stuff to it all over the place. But one of them is, I think the common name for this is sialic acid. But we take out an OH and put an amine or add groups onto the OHs, so it gets crazy. I saw these types of sugars in as components in our glycoproteins. So one of them, and I don't know for sure, but sialic acid, we had to test specifically when we were characterizing our antibodies that we made at Regeneron for sialic acid content. Because so you know that an antibody has the FAB and the FC portion, and the FAB is what interacts with the antigen. And the interaction is really all over this sugar is short sugar chains all over the structure. If the sugar chain had a sialic acid at the end, we were really jazzed because that would stop the liver from breaking down the drug. So we were looking at that. One of the tests that my group ran was to determine how many sialic acids per protein molecule that we had, and it had to be consistent. So one thing about making drugs is that the kind of drug that you made for the clinical trials, it has to be exact forever, or you have to go and do the clinical trials all over again. So the controls on making a drug are really, really tight. And it's very scary when you make new batches in a new building because things can be a little different. But we would make sure, we have to have 13 salicylic acids per molecule. So I remember doing high-performance liquid chromatography to determine that. But think recognition and when you look at sugars. All right, so sucrose, lactose, maltose, and cellulose. These are our common disaccharides. What I want you to know from these is which sugars make them up and what kind of bond holds them together. So here's the definitions of them. But really what I want to do is look at the next slide. So here's just a companion slide. So here is lactose. It's a galactose and a glucose. It has a free anomeric carbon. The galactose is this one here. And here it's a beta 1,4. Do you guys see that? Let's do some practice. This is the carbon number 1. We see this is the oxygen. Go to the right. We see carbon number 1. That's an anomeric carbon. It's up, so that's beta. It's connected to carbon number 1, 2, 3, 4 on the glucose. Some people do not have the enzyme to break a galactose glucose beta 1,4. Yes, Aaron?
If the glucose going down, why is that alpha?
The glucose, no, it's only alpha or beta if the OH on the anomeric carbon is pointing up or down. You only look at the anomeric carbon to determine if it's alpha or beta. So here, this is the anomeric carbon. It's pointing up in a weird way, but it's pointing up, you can see. And so that's going to be beta. So if you do not have this enzyme, you're lactose intolerant, and cheese pizza is not in your future unless you take the enzyme, right? Who takes those lactate enzyme pills? Some of you take those. Do they work? The tablets work better? So that's good. That's nice. They didn't have that like 10 years ago. So, or maybe they had it 10 years ago. Maybe I had a friend in grad school that had a hard time with it. They tolerate yogurt because maybe the bacteria has it. I don't know. All right, we all have maltose. Glucose 1,4 this is starch or a disaccharide. If we continued this would make starch. So maltose is a 1,4 alpha 1,4 bond. So here, this is down. It's alpha. It's connected 1 to 4. See, we just say HOH because we don't know if it's alpha or beta. It doesn't matter. Alpha or beta on this glucose doesn't matter. That's not what we're naming. All right, sucrose is glucose and fructose and an alpha 1,2. Now, what I want to point out to you guys is that I can show you these sugars and you can tell me what kind of sugar it is. Or if I ask you, sucrose, what are the sugars involved and what bond makes them up? I think that's fair. If I say sucrose, not show you the structures. How about if I do that? If I give you the name for this test, not for the final. Final might be more than that. But for this test, it's fair game for me to say the name and you to tell me which sugars are involved and what is the bond that holds them together. So for sucrose, it is a glucose and a fructose and an alpha 1,2. Cellulose is the disaccharide. Cellulose is the polysaccharide. But it's a beta 1,4. None of us have the enzyme that can break up a glucose-glucose beta 1,4. We don't. Who does? Who has the enzyme?
Cows?
Cows don't. They have friends, though, that live with them. In which stomach? Which stomach? I I don't know. They have, how many stomachs do they have? 4? That's crazy. It's really, and then what do? They throw up in their mouth and then swallow it again.
Yeah.
They chew a little bit more and then it makes it to the second stomach. That is some crazy. You don't care. Just put it next to this. I don't really care about this one. All right. So, in some of these I said what the bonds were, but I want you to know the bonds for all of them. Sucrose, glucose, and fructose, and an alpha 1, 2. And lactose intolerant, you don't have the beta 1, 4 galactose glucose. No one has this. No mammal has this, but you might have a bacteria that helps you out with that. So can they see? Then the antibiotics that they inject into the cows must not affect this, right? Because then they would all die. Then, right? But I thought they fed them all antibiotics. It's in their food, but you need it doesn't make sense to me. Does anybody understand that? I don't understand that. Certain antibiotics work on certain bacteria. Yeah. I know that there's certain bacteria that are resistant to certain, so I don't know. It's very interesting. Okay. How are we on time, guys? 9:20. All right, this is time. All right, so the polysaccharides that we're going to talk about. So when we are linking many monosaccharides together, cellulose is going to be a long chain of polymers, 2,800 D-glucoses. And it's the beta 1,4 glycosidic linkages, just like in cellulose. And again, because we don't have the enzyme that can break apart this beta 1,4, cows have the bacteria that can do that. Why is cellulose good for structure? Because we have a lot of interchain and intrachain hydrogen bonding. So when you are chewing bark, like my son when he was 4 years old would go up to trees and take bark off and chew it. He's 9 now, so he doesn't do that anymore. But when he was 4, that was his favorite hobby. And he would go to trees and chew the bark. And so what he was doing with his teeth is breaking apart these hydrogen bonds. I'm glad he's over that. Right? But again, then we can't break the bonds that will hydrolyze it down to glucose. Starch is, again, this is supposed to be alpha right here. There's a few different forms of, starch. A straight chain is just unbranched of alpha D-glucose joined only by the alpha 1,4 glycosidic bonds. This is supposed to be alpha. And so we can see this in the next slide. Amylopectin, though, is actually the same thing. It's only glucose. It is, again, the alpha 1,4 glycosidic bonds, but we have branches that are alpha 1,6. And we have different enzymes that will break up the different kinds of bonds. Amylases, ASE is for the enzymes, OSE is for the sugars, for the endings. So this amylase will hydrolyze the alpha 1,4, and debranching enzymes hydrolyze the alpha 1,6. So all these question marks on this one are alpha. And I did fix it this morning, but it didn't stick. Anyway, I'm sorry. Let's take a look at this. So this is the difference. The amylose is alpha 1,4 glucoses, straight chain. Amylopectin has the alpha 1,4 I'm showing down here and up here, although occasionally we have an alpha 1,6. These are branches. Now I say that this is a straight chain. On this straight chain, how many reducing ends do we have? One, right? So one at the end, wherever it stops. How many non-reducing ends do we have on this amylose? One, right? We have one non-reducing end and one reducing end. On amylopectin, how many reducing ends do we have? One. How many in this one? How many non-reducing ends do we have? Two. Okay. And there's more branches. Okay. So if you look back here, there's a lot of, it's branched a lot. So we have a lot of non-reducing ends. So we said that amylose was a straight chain. Really, it's not straight. It forms a coil. And at your next party, you should go to a CVS and get a tincture. It's a bottle of tincture of iodine. And then you should drop the iodine on all the food. And then you can point out to your friends which foods contain starch and which ones don't. Because the iodine molecules will get trapped within the amylose starch and turn from purple to black, right? So they might kick you out of the party. But this is something I might do at my next party. So isn't that cool? Look how big iodine is to carbon. Isn't that weird? We use this in analytical chemistry as an indicator. The starch will be an indicator for free iodine. So it goes from, if you have free iodine, it's like totally black. And the second that the free iodine goes away, it goes clear. Isn't that fun, guys? It's like the best one. So cool. Anyway, glycogen is also a starch. However, and it's the same thing as our amylopectin, except that it's more frequent branching. Every fourth or every eighth residues. So it is more highly branched. So amylose is just the 1,4, alpha 1,4 linkage. Amylopectin is alpha 1,4 and alpha 1,6 to give more non-reducing ends. Glycogen is the same as amylopectin, except it has more frequent branching. It's stored in the liver and muscle cells. And the way we have our sugars is the reason why we have the more non-reducing ends is because the enzymes to free the sugar to go through metabolism take from the non-reducing ends. So the more non-reducing ends we have, the faster mobilization we can get. So if we just have amylose, we have to chomp. We only have one place to chomp, okay, to get the glucoses off, to get it through glycolysis. However, if we are storing glycogen, we have lots and lots of non-reducing ends. So there's lots of places for the enzyme to chomp the glucose off. Glycogen, is anybody a long-distance runner? Yeah. How long does the glycogen last when you run? It lasts, for me, about an hour 15 minutes. When you run, you usually have about an hour and 15 minutes of glycogen in your muscles. And that's why, do you ever see the goo stuff? That's what people are, when they eat the goo. I do gummy bears, and I just hold them in my gums to just give a little bit of sugar so that I can get, so that means you've used up all the glycogen in your muscles. That's a good thing.
Run
But that's,
for an hour and a half.
yeah.
So
Yep.
it'd be different from like other diseases.
If you're running faster, but at a normal pace, it's, I think, yeah. For me, it's an hour and 15 minutes I hit the wall. I don't know. Maybe better people can store more glycogen. I have no idea. But for me, it's an hour and 15 minutes. I'm like, I'm done. So, but then you get over it when it starts to get it starts to go for fat. So that's what you're doing. When you only run for a half hour, you're just using up a little bit of your glycogen. But it's good to, get some of it. But why do we store glucose as glycogen? Why are we doing it? Not only just to get more non-reducing ends, we have to actually expend energy to get glucose molecules in there. And so don't look at the answer, but why do you think? The answer's in your notes, but why would you think? Well, one thing about the glycogen molecule is that it's so compact that it's not soluble. So it's insoluble, and it takes it out of creating osmotic pressure inside the cell. If you had all those sucrose or glucose molecules dissolved in your cell, it would burst the cell because of osmotic pressure. So it keeps it insoluble, so it doesn't add to the to that effect. All right, I'm just going to go quickly through other polysaccharides that I don't really care about. So here, chitin is some hard stuff that when you step on a bug, it goes crack and crunch, right? So, like if you, does anybody like to eat lobster? No? Come on, lobster's great. Anyway, chitin is a repeating disaccharide of, what we say, N-acetylglucosamine, and it really produces a like an exoskeleton because of some hydrogen bonding. And this is supposed to be beta and beta. I apologize, beta and beta here. So it's just like cellulose in plants. The chitin is just like our cellulose in our plants. In bacteria cell walls, they have heteropolysaccharides. What we mean is that you have repeating units of what we call NAM and NAG. So these guys are crazy. They have the nitrogen on there, and these crazy groups here. And then look how crazy that gets. That's unbelievable. But what happens is, and the reason why bacterial cell walls are so strong is because we are cross-linking. So here is our sugar chains, and they are cross-linked with amino acids. That is super, super strong. It's very, very hard to open up a bacterial cell wall because of this. So this is a good question that I might ask at the end of the, I don't know, why are bacterial cell walls so strong? You would answer, you not only have the polysaccharide chain, but it's cross-linked with amino acids. Plant cell walls is mostly cellulose, right? Like that, but they also contain pectin, and that's alpha right here. This is alpha. So pectin draws water in. It has an acid here. This is the bottom. This is carbon number 6 that has turned into an acid, so it's a uronic acid. This is a gelling agent in your yogurt and jams, okay? So this is, and it contains the goo in between the plant cells. The plant cell walls also have lignin that is a polymer of an alcohol. All right, so I just quickly went over theirs. I told you the one thing that I really wanted you to know. Glycosidum aminoglycans are a group of polysaccharides that play a role in cell functions. You have disaccharides repeating where there's an amino group and the other has a negative charge. A few examples are heparin, hyaluronic acid, chondroitin sulfate, right? Connective tissue. That's why people might take supplements of that. And so they're relatively inflexible, these glycosaminoglycans, and very charged. So look at the name. So glycan, it means polysaccharide, right? Amino means amino acid. So you know that there's some sort of combination. So the amino sugar, that has the negative charge and it has a sulfate or a carboxylate group, and that will draw water towards it. So your connective tissue needs that water. And of course, the hyaluronic acid, that's in the goo in your eye, lubricating our fluids of the joints. Glycoproteins, I already talked about that. Just remember, okay, so we take a look at this. Glycans is the last word, so that means there's more sugar than amino, right? Here, glycoproteins, there's more protein than sugar. So the glyco, that means sugar, and the protein, obviously we know is amino acids. So here we have more protein. So whatever's at the end, we have more of. So here, antibodies are glycoproteins. I already talked about how I would characterize the short chains. And they're the antigenic determinants. And blood groups were the first ones to look at it, to see that we saw were the sugars identified were part of the recognition. And so we have A, B, or O, depending on the sugar on the surface of our red blood cells. And so we look at the terminal monosaccharide, and that determines on the non-reducing end, and that determines your blood group. So we attach it at the end. So here, these are, don't worry about this part. I just wanted you to see if you are type A, then at the end you have a beta N acetylglycosamine. And if you are a B, you have a galactose. So that's recognition. So the take-home part of this, we make our polysaccharides, our disaccharides, but we also use them for recognition. I'm going to not show this now.